Hi all, today I am talking about the reproduction in Cycas. In my previous video, I have clearly mentioned about the morphological and anatomical features of the gymnosperm Cycas, and now I am talking about the reproduction in Cycas. Cycas is a dioecious plant, that is, the male and female reproductive organs are produced on different plant. The plant producing male reproductive organ will be a male plant, and the plant producing the female reproductive organ will always be a female plant. There are two types of reproduction in the case of Cycas. They are the vegetative reproduction and the sexual reproduction. First one is the vegetative reproduction and second one is the sexual reproduction. Now, first type, the vegetative reproduction. The vegetative reproduction in Cycas is taking place through the formation of bulbils. The bulbils are produced on the stem of mother plant. That is, the male plant will always be producing the male bulbil, and that male bulbil will grow into a male plant, and the female plant will be producing the female bulbil. Female plant will always be producing a female bulbil and will grow into a female plant. Here you can see a bulbil attached to the stem of a mother plant. See this. This is the bulbil. This is the bulbil portion, and here you can see the new leaves with the circinate vernation. Already I have mentioned about the circinate vernation and all. Here you can see the young leaves with the circinate vernation. Once this bulbil is getting detached from the mother plant, it will grow into a new plant. Now, coming on to the sexual type of reproduction, in Cycas, the sexual reproduction is oogamous type. And what is oogamy? That is, when a non-motile egg is getting fertilized with a motile sperm. This type of reproduction is known as oogamous type of reproduction, and in Cycas the reproduction is oogamous type. Now, coming on to the male reproductive organ. The male reproductive organ in the Cycas is developed in the form of cone. See this. This is the cone. And this cone will be produced at the apex of the plant. You can see where the cone is attached on the male plant. See this. It is attached on the apex of the male plant. After the development of a cone, the plant will again grow in a sympodial manner. Sympodial manner. That is, it will push the already developed male cone to one side and a new meristem for the development of the leaves or the growth of the plant is produced, and this type of development is known as sympodial development. Now, coming on to the structure of a male cone, the male cone carries numerous microsporophylls. numerous microsporophylls, and these microsporophylls will be arranged on a central axis. That is, the cone is carrying a central axis. Cone is carrying a central axis, and on the central axis, numerous microsporophylls are spirally arranged like this. Numerous microsporophylls are arranged spirally on the central axis. And the structure altogether is named as a male cone or the male strobilus. Here you can see small boat-like structure. See this. These are the small boat-like structure like this. And these are the microsporophylls, and these numerous microsporophylls are arranged spirally on a central axis in a very compact manner, thus attaining this oval-shaped male strobilus. Now, coming on to the structure of microsporophylls. See this. This is a microsporophyll. This is a microsporophyll, and this microsporophyll will be having an adaxial and abaxial surface, that is the lower and upper surface. This is the adaxial surface, upper surface, and this will be the lower surface. The male spores are produced inside the sporangia. And the sporangia in turn is developed on the abaxial surface of the microsporophyll. That is, the sporangia will be produced on the abaxial surface, that is the lower surface of the microsporophylls. You can see numerous bead-like structures. Say this, bead-like structure, and these bead-like structures are known as, small bead-like structures are the microsporangia, which in turn carries microspores. Now, coming on to the structure of microsporophyll. Microsporophyll, I have told you that it is a boat-shaped structure, and it is having a sterile and fertile region. The apex will always be a sterile region, and small lower portion will always be a sterile. The upper portion, hook-like upper portion, is known as apophysis. Is known as apophysis. A-P-O-P-H-Y-S-I-S. Apophysis. The middle portion is the fertile region. This is the middle portion. That is, the upper portion and lower portion will always be sterile, and the middle portion will be fertile. That is, only the middle portion will be carrying the sporangia. The numerous sporangia will be attached on the lower surface of the microsporophyll, and few sporangia, that is 1 to 6 sporangia, will be arranged in a definite manner, producing the sorus. See this, sorus, and this structure is known as sorus. And each sorus carries 1 to 6 microsporangia. Here you can see the structure of microsporangia. It will be hanging on the lower side of the, that is abaxial side of the microsporophyll. Thus, numerous such microsporophylls are arranged in a spiral manner on a central axis to form the cone. Now, coming on to the female reproductive structure in Cycas, the female reproductive organ in Cycas is known as ovules or megasporangium. The megasporangium in turn is produced on megasporophyll. Megasporophyll. The female reproductive organ is the megasporangium, and the megasporangium in turn is produced on megasporophyll. Here you can see numerous megasporophylls. See this. These are the megasporophylls. Numerous megasporophyll. Here also numerous megasporophyll. And these small ball-like structures are the megasporangium. The megasporangium and the megasporophyll is not arranged in the form of a cone. Each megasporophyll will be free and will be attached on the surface of, on the apex of the female plant, alternating with the leaves. Once the development of megasporophyll is over, the plant will again grow in a monopodial manner. That is, the meristem will be continuing the growth. Now coming on to the structure of megasporophyll. Here you can see different megasporophyll produced by the different Cycas species. The megasporophyll produced by Cycas revoluta, megasporophyll produced by Cycas rumphi, another megasporophyll produced by Cycas circinalis. Here you can see a small difference in the structure. Only in the morphological structure you can see some differences. And all the megasporophyll will be carrying an apical sterile portion and a lower small sterile stalk. Here also a stalk and a apical portion. See this. This apical portion is known as apophysis. Apophysis. And the megasporangium is arranged on the fertile region of the megasporophyll. See this, small bowl-like structure. The attachment of the ovule or the megasporangium will be opposite or subopposite. See this, subopposite arrangement, or it carries an opposite arrangement like this. This is all about the structure of, the morphological structure of a megasporophyll carrying the megasporangium. Now coming on to the structure of an ovule or structure of a megasporangium. The megasporangium will be carrying a central portion, this cellular region, central portion called as female gametophyte. This region is known as female gametophyte. The female gametophyte will be having archegonia. See this, these are the archegonia towards the upper side. The female the megasporangium will be orthotropous. That is, the micropyle and the chalaza will be on the same line. See this. This region is known as the micropylar region, and this region is known as the chalasal region, and even it is on the same line, this is known as the orthotropous arrangement. In Cycas, the ovule is orthotropous. Already I have told you, it will be having a centrally located female gametophyte and the archegonia. The archegonia in turn will be carrying the female nucleus. See this, these are the eggs. Egg. The female gametophyte in turn is covered by the nucellus. See this layer is known as nucellus. Nucellus, Nucellus, nucellus. And nucellus is always nutritive in function. Nutrition, supplying nutrition to the developing egg is the function of the nucellus. Nucellus in turn is covered by an integument. See, this portion is known as integument. Integument. The integument of Cycas will be having 3 layers. An outer fleshy layer and an inner fleshy layer. This is the outer fleshy layer and this is the inner fleshy layer, and a central stony layer. See this central stony layer. The function of the integument is the protection of developing female gametophyte and the archegonium. The integument open outside through a small opening known as the micropyle. See, this region is known as the micropyle. Integument open outside through a small opening known as micropyle. The micropyle is in the form of a beak. See, like this. This space is known as micropylar canal. The lower side of the micropyle will be having a small chamber, this region. Small chamber. This chamber is for the collection of the microspores or the pollen grains. So, this is known as the pollen chamber. Once the development of ovule is completed, the nucellus tissue at the archegonium, this region, the nucellus tissue above the archegonium, this region, will degenerate, forming an archegonial chamber. This region is known as archegonial chamber. This is the pollen chamber and this is the archegonial chamber. This is all about the structure of female reproductive organ, megasporangium. See this. You can see here, you can see these are the archegonium. This is the archegonium, and archegonium in turn is having a neck cell and a venter. This lower bulged portion is known as venter, and venter will be carrying the egg. This is the egg, neck and the venter, and venter in turn will be carrying the egg. Here you can see the degenerated nucellar tissue. See, this is this one, and called as archegonial chamber. In the archegonial chamber, you can see the germinated pollen grains or microspores. Above the archegonial chamber, this portion is the pollen chamber, and the integument open outside through the micropyle. This is all about the structure of megasporangium. Now, coming on to the pollination in Cycas. The pollination in Cycas is anemophilous, that is pollinated by the wind. Anemophilous type of pollination, that is pollinated by the wind. Here, the pollen grains are released at a 3-celled stage, and the pollen grain will be slightly conical in shape, like this, and is released in 3-celled stage. That is, the pollen grain will be carrying 3 cells inside. The first cell is known as prothalial cell. This one is known as prothalial cell. The just above the prothalial cell it will be having generative cell. Generative cell, and the upper cell is known as tube cell. And in this three-celled stage, the pollen grains are liberated. This pollen grain will be having an outer thick exosporium, that is the wall, thick exosporium, and inner intine. And once the pollen grains got liberated, it will deposit in the pollen chamber, where it starts germinating and fertilizes the egg. Here you can see, once the pollen grain reaches the pollen chamber, once the pollen grain reaches the pollen chamber, it will germinate and the fertilizes the egg. The generative cell in the pollen grains, the generative cell, the cell in the middle, the generative cell in the pollen grain is dividing and developing into the sperms. The sperms will be conical in shape and will be having the spirally arranged flagella. Spirally arranged flagella. The sperm are conical in shape and will be having spirally arranged flagella. See this, like this. Having spirally arranged flagella. This is the structure of sperms. Once it fertilizes the egg will develop into a zygote, and the zygote carrying ovule directly develop into a seed, and the seed germinates into a new cycas plant. Here you can see a seedling. See this. This is a cycas seedling, and the seedling is with a new leaf having circinate vernation and all. This is all about the life cycle or the reproductive structure in the cycas. Here you can see the life cycle of the cycas. I have mentioned in the morphological section that the cycas is sporophytic, that is, it is having diploid nucleus. Diploid, it is diploid, that is sporophytic, and the gametophytic portion is represented by the ovule and the microspores. So, in the life cycle, the cycas male plant will be producing the microsporophyll. Microsporophyll in turn producing the microsporangium. Microsporangium produces the microspores. And the female plant will be producing the megasporophyll. Megasporophyll in turn produce the ovule. The ovule in turn produces the egg. The sperm fertilizes the egg through the process known as syngamy. Syngamy. Here, the gametophytic phase is represented by the egg and the sperm. All other region in the life cycle will be the sporophytic. Once the egg is produced, again it will develop into a new cycas plant. This is all about the life cycle of the cycas. Happy learning. Thank you all.